Good morning, everybody. Welcome to this introduction to clinical trials. And tomorrow is Clinical Trials Day. So it's a very opportune time for everyone to learn about clinical trials. So first of all, what is a clinical trial? Well, it's a bridge between research and new treatments. And these are carefully designed and controlled medical research studies to actually see, A, is it safe? And B, is it effective? But remember, new treatments can be very dangerous, like gene therapy and stem cells. If they're not properly controlled and very well researched, they can actually cause harm rather than good. And safety and efficacy are critical issues, and that's tested very thoroughly in clinical trials because we need to protect patients. And we have to make sure that all our patients, and particularly our members in Retina South Africa, have access to these new treatments. So what are the steps of clinical trials? Well, somebody starts with a bright idea. Some person in a remote laboratory thinks this is what's going to cure retinitis pigmentosa or Stargardt's disease or whatever disease. So what then they need to do is go to the laboratory and check out. They'll look at the substance, see how they can actually manufacture it, how does it work, and then the next step is they go to disease in a dish. So we're very lucky with stem cells. Not only were they designed to treat different diseases, you can also make a disease in a dish. You can take skin cells, hair cells, blood, and you can actually take that specified cell that's already been turned into a blood cell or a skin cell and take it back to the stage of stem cells. And then you can reprogram it back into a cell in a dish that's actually got the disease or it's got the genetic malfunction. And then they can test their theory on disease in a dish. And if it works, and they've got what they call proof of principle, very often then they can actually go to animal models. Very often they go to mice because mice have a very short life cycle. And then you can actually see, is it working? If that proves safe and efficacious, then they can actually go to the trial regulators and they can apply for a phase 1 clinical trial. And that is normally done on very few patients. And those patients need to be near the clinic where they're actually doing the clinical trial because they have to see, especially if it's something like an intervention, like genetic therapy or stem cells, they need to closely monitor those people. That is normally a year or 2 years. And then if that is really successful in treat, they can go back to the authorities, the FDA in America, for example, and say, here are the results of our phase 1 clinical trial. And now we're applying for a phase 2. And if their results are good, they will be given permission to go to use more people in the trial. And they often start looking also at dosage. What is the lowest dose that has the maximum effect? Again, after 2 years or so, they can go back to the authorities and say, look, these are our results. Everything is panning out very well. And then they can probably get permission to go to a phase 3 clinical trial, which is where more people are used. Now, in rare diseases, this is an opportunity for people like Retina South Africa to get involved because they very often go globally to look for the number of patients. So if you have a rare disease, you should have a genetic diagnosis, first of all, and then you should also be registered on my Retina Tracker. And of course, you will be registered on Retina South Africa's clinical trials database. Once you've got those phase 3, and they really are very thorough at this stage, then you can apply for registration. And then, of course, you can treat many people. So that can take up to 10 years. So what about outcomes? How do we know if a clinical trial is safe? So first of all, you can have a look if there are no adverse events, and is it effective? Now that is it effective is pre-specified. And that is the problem with many clinical trials. You have to say, we're going to try out this therapy. And if people get improved visual acuity, that is the test that you do by looking at lines on an eye chart, that's visual acuity. If there's an improvement in that, that's going to be one of our outcomes. Or perhaps, is it field of vision? If people have an improved field of vision or the loss of vision of side vision slows or stops, that might be an outcome. Also, what do the ocular coherence tomography pictures look like? Those are very deep scans of the retinal cells. Or, for example, in Stargardt's disease, one of the features of Stargardt's disease is these build-up of lipofuscin in the retinal pigment epithelial layer, and those lesions can be measured. And if with the treatment those lesions get smaller or they don't grow, that might be already a sign. And the newest one, of course, is the navigation of low luminance maze. They build a maze and people can try, they look before the treatment, can they navigate this maze? And usually they don't navigate it very well. And then after the treatment, they navigate the maze and then they start to lower the lights. And of course, the lower the lights, the more difficult it is. And that is often also an outcomes measure. And then we have got patient-reported outcomes measures. And more, these are being taken into account. What does the patient feel? Does the patient feel it is helping? Can they say, yes, I can do this, I can put toothpaste on my toothbrush? That might not be important to someone who doesn't have a visual problem, but that is an outcomes measure. That can make a huge difference to somebody's life. Or I can now see the controls on my stove. Those small visual acuity or peripheral or general vision tasks are now being considered for inclusion into outcomes measures. And again, as I said, we want the lowest dose that is effective. So what is a phase 1 clinical trial? We said on approval of registration from the authority, and it's small numbers, and they need to limit the variables. If you are having a trial to test retinitis pigmentosa, you want to know that everybody in that trial, probably their disease is going to follow a similar pattern. So that's why they will choose people with a specific genetic diagnosis, which normally follows a similar pattern. They sometimes take older patients because the risk they lose their vision, it's not that much vision to lose, but they won't put, for example, children on a phase 1 clinical trial where the odds of risk are much higher. You have to, as I said, be close to the clinic, and you need frequent monitoring. And also, is there an emergency? If suddenly you start to develop a terrible infection in the eye, they need to know. And sometimes the cost is also included in phase 1. If you have people close to you, clinical trials are completely paid for, so you don't want to be flying people from country to country. You want people that are close to you that can get to the clinic easier. So that's why they choose probably people in the same town or the same area. It can go to larger numbers. Normally, they also like them quite close to the clinic or at least within the same country, because again, there's frequent monitoring, perhaps less frequent than in phase 1, but it still could be a monthly or every 2 months. And then they will start to look at younger patients. And then it goes again, they will start looking at lowering the dose to see if they still maintain that desired outcome. Then we go to phase 3 on approval again, and this is where they really start to look at larger numbers. And sometimes it's at a multi-center level, and then we have been involved in a global multi-center trial in the past for Stargardt's, and non-invasive treatments are less stringent. If there's no injection involved, if they are injecting, for example, a gene therapy into a very particular spot in the retinal pigment epithelium, that has to be reproduced. So you can't go all over the world and hope people are going to hit the right spot. So where it's a drug therapy or something like that, it might be easier to go to multi-center trials. And then children are often included if it's less invasive. But again, we want to look at the long-term effects. Now, if all those outcomes are met and everything is safe and it's efficacious, then we can go to a registration. And even at this stage, the authorities might call for an extra trial to check a particular thing that they're worried about. And this is here when we get the big pharma companies involved. And remember, every time a trial changes hands, somebody's making money, so the cost goes up. The application then has to go to the regulatory authorities. In America, it's the FDA. In Europe, it's the European Medical Association. South Africa, it's the health product regulators. And an extra trial, as I said, may be compulsory. International collaboration on licensing is often made. For example, a company in the States may hold a license to have a particular treatment. This happens in AMD, but in Europe or in Africa, they might sell that licensing to somebody else. So in South Africa, in our public health system that we know is underfunded and very overstrained, when these trials become treatments, we're going to have to assure access, and it's going to be a huge challenge to us to advocate to make sure that every South African has access to the outcomes of clinical trials. Well, what are the outcomes? What are the timelines? We know that a clinical trial can take 10 years, but luckily there is a system in Europe and in America to fast track. For rare diseases particularly, there are special designations that they will give if it's a very rare condition and it's devastating, like in LCA. They do have these inducements also to pharma. They might give them longer patents. And for example, the AIPL1 gene, that's for Leber's congenital amaurosis, it went directly from the laboratory to patients on a very special allocation from the British authorities. And I'd like to introduce you to a very special young man whose name is Chase. And he was one of the recipients of the gene therapy for LCA4. And he was born blind and he now can see. He can play soccer, he plays soccer with his dad, he can read large print. And this is really one of the rare instances where the authorities, the laboratory, and the pharma company have all brought together this special treatment because these children have to be treated before the age of 4. So how do our members get into a clinical trial? Well, I need to warn you that most trials are masked. In other words, there will be a placebo group. Now, a placebo group means people are not getting the drug or the treatment that's going to help their vision. Usually, it's only do a placebo group in phase 1. And then if they can really establish those people who've been in that placebo group for 2 years will actually then be given the treatment. So it's very important that you know that if you go into a clinical trial, you're not necessarily going to get help. You might be a placebo group. But there are also inclusion criteria, and I see I've got a lovely big spelling mistake there. And that's, because I have retinitis pigmentosa and poor vision, so you will excuse my typos. You need a clinical and a genetic diagnosis. Clinical diagnosis, we have to know that it is actually the condition that they're trying to treat. If you've got retinitis pigmentosa, you can't go there with Stargardt's disease, obviously, and hope to get included. You also might need a genetic diagnosis. And even though it's not a therapy for a specific gene, I'm trying to tell you, you might still need a specific gene diagnosis because, as we said, they want to limit the differences so that they know everybody's more or less going to respond in the same way. Your age is obvious sometimes. We know that in the Stargardt trial, for example, the MUXistat trial that we were in, people were included right up to age 50s. And then the trial did not meet its outcomes because there wasn't enough difference between the treated people and the untreated people. But, for example, in Japan, when they went and looked at the results, when they segmented out their population in Japan, the younger people actually did meet the outcomes. But overall, the older people did not respond as well to the drug. So age is more and more a criteria. And in fact, the tinlarebant trial for Stargardt's that is probably going to be registered towards the end of this year or maybe next year. It's in its very final phase 3 trial, and that has proved really successful. But they've limited the people in the trial from age 12 to age 20 because that's where they're going to see the best results. And obviously, when it becomes available, you will have to have an ABCA4 gene result to get the treatment. obviously you're going to be advised the older you are, probably the less effective it's going to be. So age plays a critical factor in all these trials. Your visual acuity may be a factor. We know that when we recruited for the MU-Exostat trial, we had to have our first criteria was visual acuity. So we wrote to our members, we recruited them, we saw everybody who had a genetic diagnosis of ABCA4, and then we said, please send us your visual acuity from your optometrist. And they had to be above a certain level of vision loss. And after that, they then had to go to the ophthalmologists and get an OCT, because the number of lesions and the size of the lesions actually was also a critical factor. And I can't comment on that because that was done under very strict criteria of privacy, and that was done by the laboratory. And then, of course, you also have exclusion criteria. You have to be treatment naive. And I keep saying to people who are rushing off to get these funny, strange treatments all over the world, please remember, it might exclude you from really effective treatment trials going forward. And again, age might be a difference. As we said today, in the Tinlarebant trial, the age was 12 to 20. It might work for you if you're over 20, but in the trial, they wanted to prove that it's efficacy in the younger patients. And your general health might be a factor that might exclude you from a clinical trial. So after all this, the outcomes is, please, get a genetic test. So let's just look very briefly at gene therapy clinical trials. That's all we're going to look at today because it's such a vast subject. We'll have to have subsequent talks. What is gene therapy? Well, it's to deliver a section of DNA or to produce a new protein, for example, in optobionics, and we'll come back to that just now. Or you want to edit out a faulty section of a gene and replace it with a working copy. And you all know how to do that in your computers. You do cut and paste, probably not every day, but certainly sometimes. That's exactly what they do in gene editing. They have a tool like CRISPR-Cas9 that can go into the cell, cut out that faulty section of the DNA and replace it with a working copy. And that is in clinical trial at the moment. This is usually gene, or even within a gene, that specific mutation only might be applied. So it's gene or mutation specific. And I'd like to introduce you to a very brave young man whose name is Stephen Howarth. He was at this stage 17 in 2007. He was the first ever recipient of gene therapy for an eye condition. He had RP, and in fact, he had LCA. He had the RPE65 gene, although his clinical diagnosis was RP, it was actually really LCA. And he responded very well. And on that gene therapy, the trials went ahead, and everybody, if you've got enough money and you've got that specific gene, can go to America or Europe or Britain and get this gene therapy. So how does gene therapy work? Every living organism contains DNA. So what we want is a benign virus like this virus, which is adeno-associated virus. You can't even see a virus, it's so small, but the researchers take out the virus's DNA, and into that virus, they introduce a working copy of a gene, and then they replicate it because viruses replicate easily, and then they can inject that new copy of the virus into the retina. And for those who can see, I've got a slide up that's showing a cross-section of the eye and an injection going right down to the back of the eye into the retinal pigment epithelium, and thousands of copies of that virus containing the new DNA material is injected. And I think that's a total miracle that the cells in the pigment epithelium say, hey guys, look, we've got a new recipe. Let's make a new protein. And they do. And the people do get restored vision. So what are promising gene therapies? And this is for Stargardt's disease I've listed here. Ocugen is an important one, and that's in phase 2/3, and that's RORA. Fast Blyer are very exciting. Stargardt's gene is a huge gene. It's on chromosome number 1, but within that huge gene, there are over 1,000 mutations, and people can have any 2 of those 1,000 mutations to have the disease. And for years, they battled to try and deliver the ABCA4 gene to the retina unsuccessfully. And now what they've done is they've actually taken the gene, split it into two, and they actually put a half into two different associated adenoviruses, and they deliver both of those copies to the retinal pigment epithelium. And miraculously, a complete protein is produced, and it's really amazing. And that is now in phase 1/2, this dual vector, and it's really going to be a game changer. And there are a couple of other ones. This is a VRT. It's a different type of endpoint. And Avant-Garde is also a very promising clinical trials. And if you want to know more about clinical trials, please go to fightingblindness.org, the American Association's really excellent website, and go to clinical trials pipeline, and you will see all the lists of different clinical trials listed there. So for RP, Usher's, LCA, and other rare diseases, for gene therapy, we've got RP, the 3 different gene types, PDE6B, 6A, and the RLBP1 gene. Those are all in clinical trials. For X-linked RP, luckily there are not that many gene variations. There's only a couple of genes that cause X-linked RP, and one of them, RPGR gene. There are 3 different clinical trials testing gene therapy. But remember, for RP, there are over 100 genes causing different forms of RP. So it's quite a big challenge for researchers. For RP and Usher's, there are 3 because there are some common pathways. For LCA, the Gucci2D gene, there's 1 clinical trial. And for LCA, the RPE65, there is another gene. Retinoschisis, there are 2 different clinical trials. For choroideremia, there's 1, but there's only 1 gene. And then we've got other interesting interventions in clinical trials like RNA therapy, AON, and optogenetics. Again, if you look at that website, you will get it. So let's look a little bit at what RNA therapy is. We've said the gene therapy is actually to deliver a working copy of the gene. RNA is the messenger that goes to the nucleus of the gene and it cuts the DNA in half and selects a specific part of the chromosome. Remember, in your cell, in every cell in the body except your reproductive cells, you've got all the chromosomes. You've got 20,000 genes. And we only use about 300 in retinal degeneration. So every part of the retina needs a different program, different code to produce the protein. And the RNA is the messenger that goes to the center, the nucleus of the cell, selects and cuts a piece of the half of the DNA and takes it to the ribosome, which is the factory part of the cell where the protein is produced. And some of these are actually now looking at intervening at the RNA to make sure that it's got the right payload. So instead of changing the storeroom where the RNA has to go and deliver it from, as the truck comes out of the storeroom, they're saying, hang on, you've got the wrong load here. We're going to just swap your load. And what they're doing is they're actually swapping at that stage the message that the RNA is carrying to the ribosome. We spoke about optogenetics. Now, even if you have very severely damaged photoreceptors, the neural layer of cells between the photoreceptors and the optic nerve, or what we call the neural phase. You've got 4 types of cells. The 2 most important ones that we're looking at is the ganglion cells and the bipolar cells, where a lot of the conversion from light to electrical and biological energy takes place. And researchers say, well, if the photoreceptors are not working, why don't we intervene where those cells are? They're carrying the message to the optic nerve. And what they're doing now is they're introducing photosensitive molecules into those cells. Some are working in the ganglion cells, and some laboratories are working in the bipolar cells. And there are 2 really excellent ones. Nanoscope, and you can look it up. Just go to Google or Edge and go and look it up. Nanoscope, they are working in the bipolar cells. And theirs is already in phase 2. Bionic Sight's really exciting, and that is Dr. Sheila Nirenberg, that you might remember, that was so exciting a couple of years ago. So those are in advanced clinical trial. And this holds hope for those people who perhaps have no photoreceptors left to rescue. So I'd like to thank you for your attention. And remember, if you have any questions, please put them in writing to our head office or to me personally. We promise to answer every single question that comes to us. Thank you very much.